And I'm also particularly in debt to Dr. Jaya for making all the detailed arrangement of my trip, making me most comfortable coming all the way to New Delhi. Okay, so right, the title of my talk actually is a bit of, well, misleading. You will see why afterward. Okay, so I think for everyone in this conference, I go without saying, it's common sense that the balance of gut microbiota is important in maintaining our health and well-being. And at times when this gut microbiota is not in a good stage, then external help could be required. For example, the inclusion of probiotic may help to alleviate some of the ill effect of the imbalanced microbiome. And the current situation is such that all commercial available probiotic, in fact, are universal. Universal in the sense that the functionality of this probiotic has been demonstrated in a specific a group of people, and then shown that a probiotic effect has been observed. And it is assumed that the same probiotic would apply and would happen to anyone who consume the probiotics. So far, so good. But we do observe that in some of the cases, the expected probiotic wasn't shown and not observed. So the gut feel is that perhaps the gut environment conditions, the lifestyles, other factors could affect the efficacy of probiotics. In fact, indeed, in yesterday's meeting, two of speakers mentioned personalized probiotic. But the question then follows is that, do we have evidence to show that indeed probiotic could be personalized or should be personalized? Even if these evidence are not strong enough to be as evidence, but are there observations that indicate and suggest such? So I'm going to summarize and discuss and go through with you and see how far and where we are now in this aspect. The first of all, the gut feeling, or rather, the first thing come to mind is that the gut microbiota of people around the world probably are quite different. In fact, people suggest that enterotypes of people of different dietary preference and so on. And for that, we conduct an Pan-Asian study initiated by the Asian Microbiome programs under the umbrella of Asian Federation of Lactic Acid Bacterial Society. And the work here shown in Asia, for Asia here, okay, were done by us, and some of the study in Italy, which is mentioned yesterday, Africa and America, were done someone else. So the methods are not quite the same. Therefore, we shouldn't actually compare them strictly, but rather, but nevertheless, they give indication of the patterns and distribution of the microbiota. And here, the family profile has been indicated. I just want you to see the color first. Oh, sorry, color first. Sorry. Oh boy, what happened? All right, you can see here. Central Asia here, whether it's Japan, Korea, Taiwan, China, both include Beijing and Lanzhou, so on and so on. They are all high in orange. Orange being bacterial CAE. And it's same for European, Italy, for example, same for the USA. They are all high in orange color, which means that they are of Bacteroides enterotypes high in Bacteroides CAE. Whereas if you have a look, right, in Southeast Asia, in Southern Asia, Bangkok, Thailand, and Indonesia, and as well as that of Africa, they are high in that of the green color, which is Prevotella CAE. So, and interestingly, if you go further north, Northern Asia, to Mongolia, you can see that Mongolia is also high in green color, which is Prevotella CAE. So ultimately, basically what it says is that along the tropical, temperate regions, people are high in bifidobacteria CE, all the way from Japan, Korea, China, to that European, to that USA, whereas Southern Asia, high in Prevotilla. And Northern Asia, Mongolia also high in Prevotilla as well. The Mongolia study is in particular interesting for reason is that Mongolia are meat eater. So the amount of meat consumption of Mongolia is only second of the USA and higher than all European countries and others as well. But yet they carry the enterotypes of Prevotilla rather than Bacteroides. So this is a very interesting aspect of it. Nevertheless, this may imply that since the gut microbiota of people in different regions are different, then the ability of a probiotic to establish in this population could very well be different. And more importantly, as you can see here, that in fact there are positive correlation between some group of microbiota and negative for others. Here in this, you can see that there are two big circles here. The left-hand side one is Prevotilla, which indicated here, Prevotilla. This is Asian and that of Mongolian, Southern Asian Mongolia. And Bacteroides is here, which is the Central China, Central Asia, European and North America. And you can see here that there are other bacterial types that seem to be positively correlated with either Prevotilla or that of Bacteroides. The blue lines indicate positive correlation. Positive correlation means that, for example, the Prevotilla, when it's present, say this sulfobibrio is present in large quantity as well. And negative 9 indicate negative correlation, that, which means that the presence of such resulted in the others being low in number. And this is not surprising. Obviously, there are competition and interaction between organisms. And based on the facts that we always observe that the total number of microbes, bacteria, in the fecal samples and in respective way you collect the samples is always in the range of 10 to the power 11 per grams of feces. So that there must be competition, meaning that the present predominant of one group resulted in reduction of the other group. That's understandable as well. Okay, and interestingly, many of these microorganisms are associated with disease and well-being of us. Either they promote a disease or they serve to inhibit and prevent diseases and so on. Give for example, the presence of Prevotilla resulted in enrichment of the sulfobibrio, and the sulfobibrio is positively associated with the ulcerative colitis and then autism as well. But the implication, we don't quite know, but this is important to suggest that in this case, the population who is high in Prevotilla could have high chance of developing ulcerative colitis. Is that this correlation need to be further probed into and studied. Nevertheless, the study suggests that in fact there are interaction between microbiota, the interaction between these people in different regions as well. Okay, and then let's see. So the question is that what happened between for this microbe present in our microbiota? All right. Some gut microbiota produce substances which inhibit other microbiota, such as those hydrogen peroxide, such as that of bacteriocins and others as well. So, but the main interaction between microbiota is in fact competition for establishing inhibition and adhesion on the intestinal mucosa surfaces. As I mentioned just now, the total number of microbiota the intestinal surface can harbor seem to be a constant value. That's why in feces, the total bacterial count is always 10 to the power 11, whatever the population profile is. So this adhesion reaction could be described by simple dissociation equation. Simply means that there's a receptor on the mucosa surface, which was found to be so as well. I'm going to elaborate after that. And the bacteria in this case would adhere and release from this receptor in a dissociation manner. Right. So a bacteria which can adhere very strongly onto the receptor will stay longer, and the bacteria that is present in bigger number will have bigger chance, will have better chance, higher chance of attaching onto the receptor. Then in this case, you decide what bacteria actually is able to establish in the person's intestinal tract. That's the case, and it was still this one, in fact, verified in lab experimentally, as well as that in human study. In the human study, the diarrhea was used as a biomarker indicator. You can see we have, in fact, pulled up all the work done on diarrhea and antibiotic-associated diarrhea, for example. And you can see here that as expected, the disease relationship is a dissociation equation relationship, and being shown here as well. Being shown here as well that when the dose of probiotic exceeds a certain level, the negative cooperative effect occurs, which means that two probiotics now compete for the same adhesion site, resulted in lower frequency of adhesion. Such is the description of association equation, meaning that too much of a good thing may not be necessarily good, and this would allow us, in this case, to design appropriate dosage to be used for subject, if you know it well, of your probiotics, right? So the dosage could be designed and developed such. So as you can see here, this personalization not only refers to the use of certain probiotic itself, but also for to achieve certain function, also the dosage and the property of probiotic should be considered as well. Such is the case. And we know that the interaction between that of adhesin present on the bacteria and out of receptor present on mucosal surface involve adhesin as well as receptor, as been shown here. Adhesin on bacterial surfaces are of various kinds. It could be present in Fimbrio verticilla, it could be a protein present in the bacterial surface, and the receptor present on the mucosal surface very often involve carbohydrate, sugar mainly. I've highlighted here two in red, mannose, because I'm going to use example to illustrate the presence of the sugars. What happens is that you have a receptor on the mucosal surface, and the bacteria bind onto it. Bind onto what? Very often, the adhesin will bind onto the sugar moiety. So you can imagine if this is the case, what you eat and what is present in the probiotic preparation when you consume probiotic would certainly determine how effective the probiotic is going to bind onto the surface of your receptor. Meaning that if I have mannose present in my food matrix where probiotics present, and this mannose is going to bind onto the bacterial adhesin, resulted in the bacteria, the probiotic, not able to bind onto the receptor on the mucosal surface. In this case, it rendered the probiotic ineffective. So that's one point which we should remember. In this case, special personalization also involves the tailored design of the probiotic concoction. And you can see here again, you can see mannose here. Mannose prevent inhibition of the various gut bacteria to different extent. You can see E. coli, for example, is highly sensitive to mannose. So the presence of mannose, in fact, inhibit E. coli attachment on the mucosal surface. And whereas Salmonella is less sensitive to that of mannose. So that we should bear in mind in the design of our probiotic concoction. All right. And for the two famous probiotic most widely used in the world, the Lactobacillus casei shirota, as well as the Lactobacillus GG, you can see here that the adhesion onto mucosal surface is very much affected by which sugar is present in the preparation. Enhancement referring to positive, and then you have negative cooperation represent inhibition. Take, for example, the top one, Lactobacillus rhamnosus GG. As you can see here, galactose does inhibit adhesion, fructose enhance it, and fructose enhance it as well, and mannose inhibit, and sucrose enhance it, and so on and so forth. So the choice of your sweetening agent is important. Besides the sugar, besides being a sweetening agent, in this case, it does affect adhesion property. And also, we do know that a variable energy compound sugar, very often, the presence of sugar is desirable to provide the cells, allow it to maintain themselves in the final preparative products. So the presence of certain desirable sugar, in this case, would prolong the shelf life of the probiotic, especially in conditions whereby refrigeration is not readily available, present in a room environment condition, for example. So this is design of the probiotic itself. Right, so besides the differences of probiotic profile, or rather the gut microbiome profile of people in different regions, in different part of the world, our probiotic change with age as well. Two speakers again mentioned yesterday that probiotic change with age, and in our study, people in different parts of Asia that shows changes with age. And two most obvious changes, which was mentioned in fact yesterday as well, are the bifidobacteria, and enterobacteriaceae. So, and why do I mention that again? Of course, the physiology of the age of the people as one grow older does change. And what is the significance here? I just want to show the differences in different countries. Right. Granted that as a whole, bifidobacterium decrease with as we grow older. And we know that bifidobacteria is supposed to be beneficial microorganism, supposed to prevent diseases and so on. But you can see here that in general, you can see here that the Chinese bifidobacterium drop faster than the other group of people in Asia. Korean as well. Whereas Mongolian, the bifidobacterium doesn't drop so much, which implies that in this case, it is probably the aged Chinese and that of Indonesian and that of Korean who did supplementation of bifidobacterium, for example. So this is one example of how the specific probiotic could be used to enhance certain needs of certain subpopulation in Asia, in this case, for example. Right. On the other hand, Enterobacteriaceae increase with age. We mentioned yesterday as well. But you can see here of a Chinese Enterobacteriaceae actually doesn't change very much. The most drastic change was observed in Indonesian. So that is the case. Indonesians need some help. Chinese perhaps not so much as far as Enterobacteriaceae is concerned. All right? Okay. So then the second things that come to mind is that for different population probably have different nutritional needs. I'm citing some example. These are in fact common sense. In fact, I mean, for example, people, the lactose intolerance. And Asians certainly need more help than the European. In Asia, for example, in China, 75% of us cannot actually break down lactose, whereas European less than 15%. So therefore, the lactose degrading properties for probiotics is important, more important in Asia than European. And again, point number two, some probiotic was demonstrated to produce and deliver vitamins, for example, the vitamin B11, vitamin B2, vitamin B12, and so on. And also some probiotic bacteria are being involved in the breakdown of phytic acids, which is a non-nutritional factors which inhibit iron. And these probiotic properties are probably desired in developing country, in malnutritional developing country especially, and perhaps not so much in developed country and people who are well fed. So therefore, the different population, in this case, the developed and not underdeveloped, would require different supplementation of probiotic that show different probiotic properties. Point number three, again, as I demonstrated just now, some probiotics are negatively associated with disease-causing organisms, such as that of diarrhea and necrotizing enterocolitis. And this, of course, more often observed in developing countries in Asia and Africa, and less that of developed countries. And of course, if you are going to design probiotic, then the needs of different countries comes in as well. So some probiotics has been reported to bind and remove store toxins, which include heavy metals and aflatoxin, and able to detoxify some of the toxins. Then in this case, the non-hygienic, a country such as that of developing countries will require more help of probiotic that, in fact, able to perform this function. All in all, these are examples of nutrition needs of a specific group of people, and then in this case, the probiotic supply provided should, in fact, hopefully, or is more desirable to provide these properties. Right. Then the third category we can think of being observed is that how well the probiotic able to survive in food matrix. Very often, the probiotic today are provided in the form of couch fermented milk, of either milk or cereal or soy milk, for example. However, after being consumed, you also consume other types of food as well. And the ingredient in food affect probiotic organism. Well, the gut feeling is probably, yes, isn't it? Here, these last few days, we take Indian food, and Indian food is very spicy, especially for European. And spices, we know that spices are bactericidal. I mean, the fact that country in tropical country, places which are hot and humid, such as that of India, which is hot, such as that of Sichuan and Hunan in China, which is hot and humid, and Indonesia, which is hot and humid as well. You take a lot of spices. It's because through observation experiences, spices were found to inhibit pathogenic organism, diarrhea-causing organism, thus render the food safer. And that become a cultural tradition. And this, nevertheless, is going to affect the microorganism of our gut. It's going to affect the microorganism we take probiotic, for example. So I'm going to, there are many of these examples, red peppers, garlics, and teas, as has been shown in affecting the vulnerability survivors of probiotic. I'm going to cite some example here. For example, consumption of garlics. Garlic oil sulfide as garlic oil powder has been shown to inhibit many organisms. In particular, I've highlighted here, Bacteroides fragilis is particularly sensitive to garlic. And here again, tea polyphenol has been shown to inhibit different bacteria. And here again, I use Bacteroides fragilis example. As you can see, catechin, the second from the left, inhibit the vulnerability survivor of these Bacteroides fragilis within a certain time limits, within a certain time, or in 24 hours, by half. And some organisms are more resistant to catechin, others are less. And polyphenol in tea could not be absorbed in the intestinal tract. So therefore, they stay in the intestinal tract and eventually pass out. In the intestinal tract, some other microorganisms may break it down, degrade it, and produce other products, such as the three listed on top. These are metabolic products of intestinal microbes of tea polyphenol. And you can see here, I'm citing example of Bacteroides fragilis again. You can see here that Bacteroides fragilis is very sensitive to all three end products of polyphenol metabolism, which suggests that, which is in fact therefore not surprising that the garlic eating and tea drinking Chinese are low in Bacteroides fragilis as compared with other Bacteroides. And interestingly, when you look at the microbial profile of Indonesian, which in fact, just slipped my mind, I should include the data, but publish that anyway. In Indonesia, healthy children were found to harbor Bacteroides fragilis. among all the Bacteroides. And over 80% of Bacteroides are Bacteria fragilis. And when we probe a bit further, we found that the Bacteria fragilis harbored by Indonesian children regulate immuno properties strongly. Perhaps explain why these children are healthy, because the presence of Bacteria fragilis that help it to combat disease and develop inflammatory diseases and so on. And more interesting, we published that as well, is that we found that this particular bacteria does interfere with lipid metabolism, interfere with the fasting protein production, expression, and as well as the responsive receptors to the factors, and making the children fat. And indeed, it was the case. For all the healthy children we work on and where the bacteria is isolated, they are all chubby. So that in fact make the parents very happy. And but that also one example of how, in fact, indeed, the presence of the diet affected intestinal microbiota and microbiota, and this, in fact, the physiological stage and health of this individual. Right. The next factors, which in fact is quite obvious, has been explained and reported many times, is there was mentioned as well. The effect probiotic is strain specific. For example, the different lactobacillus being present here has been shown to have impart different immunomodulating properties of the host. Some may be immuno-enhancing, other anti-inflammatory, and so on and so forth, this example. And even also that apply to Bifidobacterium as well. The different Bifidobacterium species or strains have been shown to modulate the production of different cytokine of the host to different degree, as you can see obviously in this graph. So depending on what you want and what do you want the probiotic to do, perhaps you design and select probiotic or selected properties. So it depends on the needs of the population. You could then come up with personalized selected probiotic, which in fact is important. And all in all, we believe that the advances in technology, now it's easier and cheaper to profile microbiota, to do metagenomics, to do metabolomics studies and so on, to allow us to understand the microbiota we have and the function they can do or they are lacking. And in this case, it facilitates our design and development of personalized the probiotic. And this personalized probiotic, I must say, is not necessarily person-dependent, but rather group and subpopulation dependent. For example, certain parts of India will require certain type of probiotics, certain groups in Indonesian as a whole will require other types of probiotic, thus regionalized and personalized probiotics. And therefore, in a recent concept paper which is going to appear in Trends Biotechnology, we argue that writing is already on the wall. It's a matter of sooner rather than later that the consumer would demand personalized, targeted probiotics. And you'll be surprised that matter of times the regulator would also demand not only the edification of probiotic, also that of the functionality of the probiotic against specific targeted group. I understand that, I mean, some companies already started doing that, has already looking at their bacterial property, their probiotic bacteria in establishing different population of in the world and their functionality and so on and so forth. I think that is going to come. Okay, thank you very much.
This was an extremely thoughtful lecture on or talk on the raising very basic questions regarding the use of probiotics in different individuals, depending on the area of residence, on the age, on the diet factors, etc. I think this requires, there will be many questions, I think. Let there be. Yes, Dr. Batish, yes.
Dr. Lee, a very good presentation. Actually, I just wanted to know what are the prospects of universal probiotics, concept of universal probiotics? What is the status, Is it really, it can be, a practical approach or just on the pipeline, I mean, still a lot of work has to be done.
Right. In fact, although we discuss on personalized, regionalized probiotic, there are some probiotic property, in fact, are universal, probably what you have in mind as well. Some common properties would be desirable for all populations. In this case, these are universal probiotics. But looking at it, there are actually not many properties which are universal.
Because as far as I understand, because the efficacy of probiotic is not strain dependent, but host dependent also.
That's right.
Because it may vary from different hosts,
yes,
from different regions.
Indeed.
But if we can conceptualize, I mean, we can put into practice to develop, universal kind of probiotics, it will address all these local issues,
Sure.
Yes,
sure.
please.
Yeah. Very possible. Yeah.
Dr. Lee, your addition experiments were very interesting. So when you have chosen a mixture of strains, will they compete against the same receptors, and some will stick and others will not find an opportunity? Because in the lactobacillus species, there could be common receptors. So how do we deal with this kind of situation?
Right. Basically, get to know your probiotic well and get to know the target organism well. As you say, if the pathogenic organism and the probiotic in fact share the same receptor, then there will be competition. If they do not, then the probiotic will not have a probiotic effect in actually elevating, in preventing the action of the pathogen. But having said that, of course, probiotic has a different level of effect. It does, in some cases, some probiotic does modulate immune responses. In the case through the immune reactions, it may still prevent, inhibit, or enhance diseases as well, for example.
So will you say that addition will be one criteria of selection, or?
Yes, I agree.
It will be one criteria.
That's right. I agree. It does make sense.
Please,
that way.
yes. Please identify yourself.
Yes, I'm Dr. Anita Chaudhary from Indian Agricultural Research
Can't hear you.
Institute.
It's not on, isn't it?
Please.
I'm Dr. Anita Chaudhary from Indian Agricultural Research Institute. I just want to know, is there any kind of a high throughput assays, because there are so many strains which needs to be checked for the adhesion properties or something. Instead of using the cell lines, maybe some kind of a simulatory assays on the high throughput scales.
Yeah.
If
In fact, they are. The important thing is that you know your markers, you know the targets. Then you can, in fact, screen. You can design a high throughput screening processes. Take the most basic, fundamental one example, for example. Let's say you want to help people in breaking down to digest lactose. Then in this case, you can just target at lactase, and make screen for organisms that produce lactase. We did that, and in fact, the ability of lactobacillus or lactic acid bacteria in producing lactose is vastly different among them. So it's like whatever you want.
That boy on the last, please. There.
With regard to the binding kinetics you did, the problem we faced is the blocking the non-specific binding in the plate binding assay. So how did you resolve that? Because many of these probiotic strains bind to BSA commonly used.
Yes.
So how did you resolve that? Which block?
It depends on your assay protocol, assay systems. So that we can discuss in more detail. Basically, how do you, if it's an in vitro assay, how do you coat your mucus, for example, mucin, for example, if a cell study, how do you mask the surface where no cells growing and so on. These are the details which I can discuss with you.
And again, with the mucin, the problem is it's very complex and
Yes.
you cannot do really proper molar calculation.
It does. Mucin composition is depend on the food diet you eat. Therefore, work on your population. You cannot just buy a mucin from somewhere and assume that it's going to work for your population. Just isolate the mucin from feces of the subject.
No. My question is that mucin
Yes.
is so heterogeneous.
Sure.
So binding experiment will be problematic in terms of
Sure.
if you calculate in molarity.
Sure. It is.
please.
Therefore, you have to target it. Yeah.
Please stand. Please identify.
I am Dr. Ghosh, Jawaharlal Nehru University, Delhi. Thank you for the excellent presentations. Many questions, I'll just hone on to two. One you said, which is very significant to our country, is probiotics and spices. India is a spice-using country. Do you have, or does your consortium have specific data on how major spices like curcumin in India, for example, affect probiotics? That's question one.
Yes.
Can I continue?
Yes.
The second question is, you raised another interesting domain of having designer probiotics possibly for Asian population or even country-specific population.
Yes.
Your comments on that, please. Thank you.
Yes. Well, I think I make a mistake of mentioning spices because spices we are particularly interested in. And having mentioned that, I could imagine that many of you will start working on spices as well. In fact, spices is very not well studied at all. We did some study and we searched literature. In fact, there are very few studies on spices for simple reasons that spices is only largely consumed in Asia. And of course, many of this work was done in the West and so on. Spices was not, in fact, picked up and pay attention to. But it is very important factors which worth our systematic study and looking into it at all. And the other question of the regional design probiotic, right? It's right, that's right. In fact, there are not many data which shown that. I mean, basically what we're suggesting a direction and a trend to work on, but there's no systematic data which show such yet.